So in the same manner, there are hormones also. We know maximum hormones are made up of protein. There are few which are made up of steroids or cholesterol, you can say. Otherwise, maximum hormones are proteinaceous in nature. We have learned already regarding insulin. We have learned about glucagon. Insulin decreases the blood sugar level to maintain it to the normal. Glucagon increases the sugar level. So they work against each other, but for our better. And they both come from the same gland, which is called as pancreas. So hormones play an important role. Without hormone, body's growth and development, proper metabolic activities, daily activities done in the body are only possible because they are under the control of hormones, and these hormones are again protein. In the same manner, we have receptor proteins also. So proteins which occur on the surface of the cell membrane, and they receive the specific information molecules, like for example antigens present on the surface of a virus or antigen present on the surface of a bacteria, that means pathogenic organism. So these information molecules, and then they are activated to form defense or various cellular functions. So once these receptors receive some information, because they are receiving information, they are called as receptor molecules. They are present on the cell surface. They are generally glycoproteins. So once they receive information, they signal the cell inside with the help of secondary metabolites, with the help of secondary messengers, for example cyclic AMP. In the next class we are going to learn about this. So this helps in developing new functions or new chain of reactions inside the cell. This is how cell perceives the environment and acts accordingly. So receptor proteins are on the plasma membrane. Next is, which you will be learning in details in the 12th class, is the control of gene expression. We all know DNA forms mRNA. mRNA is the messenger protein. Messenger proteins are formed in the nucleus, and finally this messenger RNA. This messenger RNA is formed in the nucleus, and then it goes out into the cytoplasm for protein formation on the inside the ribosome, because ribosomes are protein factories. So gene expressions are also controlled by mRNA, because if mRNA is not formed, then the protein formation is not possible. So DNA, or you can say genes, depend upon RNA. And then now next is, but there are proteins which control this process, because there are some proteins which are able to cause faster rate of gene expression, slow the rate of gene expression, stop the gene expression. So this will be in your 12th class, because you will be learning about repressor protein, you will be learning about operator protein. So there are many proteins which control the gene expression. Now the next function is the structural and the support proteins. So there are proteins which form extracellular structures. They form the matrix of the connective tissue. We have already learned collagen, elastin. So collagen provides tensile strength to the connective tissues, tendons, cartilage, bones, ligaments. So elasticity is provided by by elastin to the ligament. So all these things are present. All these things are present in these connective tissues, and they are important because they provide structural support to the tissue where they are present. In the same manner, movement also, because when we talk about pseudopodia, in the 10th class you have learned about amoeba. Amoeba performs ingestion and movement with the help of pseudopodia. Now formation or withdrawal of pseudopodia, this all is controlled by microfilaments. And if you have remembered the concept, we have learned already in the 8th chapter that microfilaments are the cytoskeleton portion which are proteinaceous in nature. So microfilaments help in pseudopodia formation. In the same manner, myosin is there, tropomyosin is there. There are many proteins which form the muscles in the human body. So muscular movement, pseudopodial movement. Now come to flagella, cilia, flagella of the sperm or flagella of the bacteria, though they are made up of different protein, because flagella of eukaryotes is made up of tubulin, but the flagella of prokaryotes is made up of another protein called as flagellin. Please remember, prokaryotes will never have cilia. Prokaryotes will always have, if they have, flagella, and that flagella is made up of flagellin. Humans, or you can say eukaryotes, can have flagella as well as cilia, but remember both of them are made up of tubulin. So all these proteins, all these structures, whether you say flagellin or tubulin or microfilaments or myosin, actin, all these proteins, all these molecules are nothing but proteins. So proteins also help us in the movement. In the same manner, we have protoplasm. Now we know protoplasm of the cell. Proteins form, in the last classes also I have told that proteins exist in the colloidal nature in the protoplasm of the cell, and in the protoplasm as well as in the organelles. So they form important component of the protoplasm, that means cytoplasm plus the organelles as well as the plasma membrane. So proteins form 50%, I have told this before, 50% of the dry weight of the living content is nothing but the proteins. Then similarly there are some proteins which are protective in nature. So for example, they provide us, here protective basically means that defense on the surface. So there is a protein called as keratin. Now we all know keratin is present in the skin, present in the hair, present in the feathers, scales, nails, claws, hoofs and horn, and they all are protective structures for the animals including human. So protein play an important role even on the surface to provide us protection. And then the last we can learn is the compatibility. What is compatibility? We have all understood in the junior classes regarding pollination. When pollen grains are released from the anther, they fall on the stigma surface. Now, it could be possible an insect or wind may have carried a wrong type of pollen grain on the stigma. So, how will the stigma understand which pollen grain has come on the surface or not? So, now surface to surface, it means stigma surface cells and the pollen grain is a cell. Stigma surface is multicellular, pollen grain is single cell. So there is a single cell structure, it may carry 3 cells inside, 12th class topic. So that single cell is going to communicate cell surface to cell surface communication with the stigma, and finally stigma understand whether it is the right type of pollen grain, should it germinate to prepare pollen tube and then cause double fertilization, topic of your 10th class. So this is called compatibility. Interaction will make the pistil understand or stigma understand that right type of pollen grain has landed or not landed. If it is right type, compatible. If it is not the right type, incompatible. And finally fertilization may occur or fertilization does not occur.